Hello students, in today's class we are going to learn about plasma membrane structure and function. So coming to the functions of plasma membrane, firstly it is important in compartmentalization. So the membranes are continuous unbroken sheets and are as such inevitably enclosed compartments. They allow specialized activities to proceed without external interference and enable cellular activities to be regulated independently of one another. Secondly, they provide a scaffold for biochemical activities. So normally the biochemical activities that will take place in the solution will involve the reactants such as the enzyme and the substrate. They need to interact with each other, and these interactions will be dependent on random collisions. However, if some of the components are actually embedded in the membrane, they will provide a scaffold or a binding site for the enzymes, for example, and in this manner, there will be no dependence on random collision. Rather, the reactions will take place in a more ordered and effective manner. Then the plasma membrane are a selectively permeable barrier. They promote movement of select elements inside and outside of the cell. They will allow transport of solutes according to the concentration gradients. They will also allow transport of other molecules such as ions. Plasma membranes are also involved in responding to the external stimuli. For example, the stimuli can be anything such as presence of a particular sugar, and this acts as a signal that regulates the internal activities that will include signaling or signal transduction, which will allow the specific group of genes required for metabolism of the particular sugar be expressed. Then the plasma membrane also allow for intercellular interactions. They allow the cells to recognize and signal one another, to adhere when appropriate, and to exchange materials and information. The proteins within the plasma membrane may also facilitate the interaction between the extracellular material and the intracellular cytoskeleton. Lastly, energy transduction. So ATP generation is the important phenomenon that takes place in the membranes of organelles such as chloroplast and mitochondria. Now, coming to the models of plasma membrane, different models were proposed for the structure of plasma membrane. Some of the initial models were the Gorter and Grentel model, and then later on other models such as sandwich model and fluid mosaic models were also proposed. So we'll start with the discussion on the first model, that is Gorter and Grentel model. So these scientists were involved in investigating the surface areas of the lipids, and for their experiments they extracted lipids from RBCs of humans and animal cells in acetone. These lipids were spread on a surface of water, and with the help of an instrument which is known as Langmuir trough. Then they measured the surface area of the extracted lipid and performed the calculations, in which they divided the surface area occupied by the extracted lipid by the surface area of all the RBC membranes. Now, based on their findings, what they actually obtained was that the amount of lipids that were extracted were so much that they can wrap around or cover the RBC membrane surfaces at least twice. So based on these findings, it was being proposed that lipids are present not forming a single layer, but they are actually present in the form of a bilayer on the surface of the membranes. So that is how the lipid bilayer model was proposed, according to which the lipids are organized such that, so the lipids are basically made up of the polar head and the non-polar tails, and they are organized such that the heads are present interacting with the extracellular and the cytosolic environment, and the tails are present embedded such that they avoid interaction with water molecules. However, there were some errors to the proposed structure. First of all, acetone does not quantitatively extract all the lipids, and the calculation of the membrane surface area were also less than the actual figure. Besides, this model could not explain the surface tension and the membrane transport. Later on, the surface tension of the membranes were calculated and were found to be much lower than those of the uh, just the pure lipid structures. And this decrease in the surface tension could be explained by the fact that proteins are also present in the membrane, which were not accounted by in the first model. Hence, later on, the sandwich model was proposed by Danieli and Davison. According to this model, this was the first attempt to describe that the membrane structures in presence in terms of molecules and to relate the structure of biological and chemical properties of the molecules to the structure. So, according to the bimolecular model of Danieli and Davison, the plasma membrane is made up of two layers of phospholipids, that is, it is bilayer or bimolecular, in which the phospholipid molecules are arranged in such a way such that the hydrophilic heads of the phospholipids face outside and the hydrophobic non-polar lipid chains are associated in the inner region of the leaflet. The hypothesis also suggested that the polar ends of the lipid molecules are associated with monolayer of globular proteins. Okay, so what it basically meant was they suggested that the protein molecules are present that are sandwiching a layer of lipids in between them. Hence, the name sandwich model was proposed. However, there were some errors to this model as well, as it failed to explain the thickness of the membranes. Also, it did not took into consideration the other factors such as not all the membranes are identical or symmetrical. Membranes with different function also differ in their chemical composition and structure, and membranes are bifacial with distinct or separate inside and the outside faces. So later on, the unit membrane model was proposed, and it was concluded that the plasma membrane and membranes of all the cell organelle were actually similar in structure, and hence the generalized unit membrane model was proposed. The unit membrane visualizes cell membrane as trilaminar in structure and indicates that the structure consists of two dark or the osmophilic layers separated by a light osmophilic layer. The physical appearance of the trilaminar model had led to the use of the term that is known as the unit membrane. The unit membrane complex implies that it is trilaminar appearance, which is made up of two protein layers that are sandwiching a layer of a lipid in between. So each dense osmophilic band is made up of a 20 angstrom thick protein layer, and the polar heads of the phospholipids are approximately 5 angstroms, and thus the dark band will form about 25 angstrom thick layer. And the clear osmophilic zone is about 35 angstroms in thickness, and it is bimolecular layer of lipids without the polar heads. In other words, you can say the unit membrane is 75 angstrom thick. The 35 angstroms thickness is of the phospholipid layer between two 20 angstrom thick protein layers. So in unit membrane model, the protein layers are asymmetrical. On the outer surface, the proteins are basically mucoprotein in nature, while the inner surface proteins, they are non-mucoid proteins. Later on, the fluid mosaic model was also proposed. According to the fluid mosaic model, this model was able to explain the fluid nature of the membrane, and it stated that the membrane is not completely rigid structure. It is basically flowy or fluid in nature, and it is the main structure consists of a lipid bilayer, And then two different types of proteins are associated with the membrane. The first type of proteins are known as the integral membrane proteins, also known as the transmembrane proteins, that traverse through the lipid bilayer. And second category of proteins are the peripheral proteins, which are attached on the surface of the lipid bilayer, either towards the inner surface or towards the outer surface. Then we will also find presence of certain cytoskeletal components towards the inner side, attached to the inner surface of the lipid bilayer. And we will also find some embedded cholesterol molecules within the, according to the fluid mosaic model. Now, the fluid mosaic model basically describes the structure of the plasma membrane as a mosaic or a network made up of phospholipids, cholesterol, proteins, and carbohydrates that gives the membrane a fluid-like character. It is about 5 to 10 nanometers in thickness, and the proportion of these different components will vary depending on the cell type. Coming to the fluid quality of the membranes, it was proposed. So this model was proposed by Singer and Nicholson, and the membranes are held together by the hydrophobic interaction. Most membrane lipids and some proteins can drift laterally. That is, they can, within the membrane, they can move from left to right, but rarely they can flip transversely. Means the proteins or molecules that are present on the outer leaflet will not flip onto the inner leaflet. Then some membrane proteins are also attached to the cytoskeleton, and they cannot move much far. And lastly, the membrane must be in, should have a fluid character to be functional. So the membranes are basically mosaics of structure and function. The two different types of proteins that are present in the membrane, as we just saw in the diagram, the integral membrane proteins and the peripheral proteins. Integral proteins are those that are more strongly associated with the bilayer, with the hydrophobic and the van der Waals forces. And they can also be said as transmembrane proteins. Peripheral proteins, they are not embedded, but they are attached to the surface of the membranes. On the cytoplasmic side, they may be held by filaments of the cytoskeleton. Then coming to cholesterol molecules. So cholesterol molecules, they function as a fluidity buffer. So these cholesterol molecules are embedded in the animal cell and not in plant cell membranes. Uh, and they make the membrane impermeable to water soluble substances. It also stabilizes the membrane, that is, below the melting temperature of the membranes, the cholesterol will disrupt the close packing of the fatty acid chains, and thus they will help to increase the fluidity. On the other hand, if the temperature of the membranes is increased or the cells is increased, then the cholesterol molecules will restrict or constrain the movement of the fatty acid chains. In this way, they will decrease the fluidity. And hence, cholesterol molecules are basically known as a fluidity buffer, so that they can modulate the fluidity of the membrane and make it more stable. The membranes are also asymmetric in nature. The two layers, that is the outer leaflet and the inner leaflet of the lipid bilayer, they may differ in the lipid composition and also in the composition of the proteins. They may also, they will definitely also differ in the presence of carbohydrates. So carbohydrates are majorly present in the outer membrane itself. So this asymmetric orientation of the plasma membranes or the membranes begins during their synthesis of the new membranes within the endoplasmic reticulum itself. Here we can see the representation of different types of lipids that are present in the membranes. And as you can see, for example, phosphatidylethanol is present as a major component of the inner monolayer then compared to the outer monolayer. some others such as phosphatidylcholine, more of the phosphatidylcholine molecule will be on the contrary present in the outer monolayer compared to the inner monolayer. Thus, lipids are asymmetric in distribution. Coming to the conclusion, plasma membrane provides a barrier between the inside and the outside environment of the cell and plays an important role in cell-cell signaling and communication. It is composed primarily of phospholipids and proteins, and the exact composition of the cell membrane varies depending on the cell type. Although there is a wide variety of cell types which exist in nature, one uniting feature that they share is their unit membrane nature. So that's it about structure and function of plasma membrane. Thank you, and kindly like, share and subscribe if you like the content.